actually a continuation of a project that I had started a couple of years ago, and it's related to the spindle assembly checkpoint, which is a mechanism within cells that regulates cell division. So we're talking about the transition from metaphase to anaphase in the cell cycle, which is wherein the chromosomes are separating to create new daughter cells, and it's really important that they separate correctly, otherwise you get developmental diseases, or that's how Down syndrome happens, and other things. So I'm studying this pathway that triggers a halt in the cell cycle between metaphase and anaphase if anything's wrong with the chromosomes. And so if there is anything wrong, it'll stop the cell cycle, and then repair mechanisms can come in and fix whatever is wrong with the chromosomes before the cell cycle is allowed to continue. So I study in particular BUB1, which is a gene involved in the pathway, and it seems like it's at a pretty important part in the pathway, so I decided to hone in on that one. It's been conserved in yeast and reptiles, and that's about it. So I decided to study it in fish because it just hadn't been shown before. So last summer I did some work and showed that it was indeed conserved in fish species, two different species that I had tested. And then I treated the oocytes that I collected from the fish with different cellular treatments to compare the expression patterns of the gene within these cellular treatments. So the first one I did was progesterone, which sort of emulates a normal cellular condition, and found that BUB1 was expressed more highly in a cell that had that normalized treatment than as compared to a control. And then this summer I treated the cells with microtubule destabilizing drug, nicotazole, and what that does is it depolymerizes or shortens microtubules, which makes them, microtubules normally attach to the chromosomes and help push and pull them apart for cell division, but if they are at all destabilized, they won't have the right tension to push and pull, and then bad things will happen during cell division. So treated it with this, and then from there we can assume that the cell might be under arrest in between metaphase and anaphase. And at this point I detected for the expression of BUB1 by extracting RNA from the oocytes themselves, and then doing a reverse transcriptase polymerase chain reaction, which is a pretty established lab technique, to convert it back to DNA, and then ran a DNA gel to detect the expression patterns, and found that the gene transcript of BUB1 was more highly expressed under these destabilizing conditions with plus nicotazole than they were in the control. And this is now statistically significant with good p-value. So what that means for the overall body of knowledge is that BUB1 might be involved in this pathway in a manner that responds to abnormalities with the microtubules, and so I would like to look into that further. So what I did to look into that was I planned on taking microscope images of the microtubules and chromosomes themselves to see if there's any morphological damage. So the only picture I got was this, which is not under any sort of destabilizing treatment. It's just a normal follicle cell. And so from there we can go to do immunofluorescent assays, which would detect if protein is actually in there and where it's localized, if it's localized around the microtubules, if it's next to the chromosomes themselves, and that'll help us further eradicate what the role of BUB1 is in this process.